Swayam Prabha. Digital India. Educated India.
Okay, we'll carry on with from where we left off. So, last time I think we stopped at equation number 10, right. We were looking at this flux due to both potential and concentration gradient. So, what we started from in that case was we started from this equation where J of P, right, is equal to minus d del C over del of x and then minus Z E C U del psi over del of x, and this equation, well, I forgot what the number was, but anyway, this was the equation we started with. What I forgot to mention at the time is this equation is referred to as the Nernst Planck equation. Okay. So, this equation is popularly referred to as the Nernst Planck equation. Now, equation number 10 actually went like this. I will just show what equation number 10 was again. So, this was equation number 10 where we stopped last class, where it says that the flux of the charged particles varies as this, where the first term in the bracket is del ln c over del of x with the corresponding constant, and the second term is del psi over del of x with the corresponding constant, right. The first one concentration gradient, so you know the constants of K and T, right, that is what comes from chemical potential. The next one is the potential gradient. So, obviously, if you have a potential, you are going to have the constants which are respectively the valency and the corresponding charge E. Okay. So, that is really what matters. Now, how can we simplify this further? To simplify this, to simplify equation number 10, what can we do? So, again, as we also briefly discussed last time, your mu electrochemical potential was given can be written as mu 0 plus k T ln C, right, plus Z E times psi. Now, we can write this. The first part is mu, just mu because of your concentration, and the next part is what? Because of the potential, right. Now, if you are going to do del of x, where mu prime obviously is your electrochemical potential, then what we will have is the first term will go to where? 0, right, because it is a constant. So, we will be having k T del of ln C over del of x, right, plus Z E del psi over del of x, and let this be equation number 11. Okay. So, we take this and we use 11 in 10. So, using 11 in 10, you will see that it simplifies to a very simple equation. That means, I can write J C is equal to minus Z E C U del of mu prime del of x, okay, and this would be say equation number 12. Now, this makes sense again, because remember this J C is the flux of your charged particles which are not only moving because of a concentration gradient, it is also moving because of its potential gradient. So, it just makes sense that the final J c, right, and that is what we are describing, it is a combined flux, the J c would only depend upon the gradient of your electrochemical potential. It is not the only the electrostatic, neither the concentration, it is a combination of both, because in a system you would be having both of these coexisting at many different times. Now, having said this, remember we also I also told you that we derived Nernst equation before from general thermodynamics, right, and we got to the Nernst equation. Can we get the Nernst equation from here? If so, how can we get it? Now, the thing is very simple. The thing is, if you are considering an equilibrium scenario, that means consider equilibrium, that means it has equilibrated with respect to both your chemical potential and your electrostatic potential. So, what will happen to the flux J c? It will be 0. So, consider equilibrium, then J c would be equal to 0. So, if J c is equal to 0, from 12 what do we have then? See, Z, E, C and U, these cannot be 0, right, these are constants, right. So, what we can have is del of mu prime del of x would be equal to 0, right. So, this is del of x, my x almost looks like n. So, this is 13, great. Now, do you think we can get Nernst equation from here? You look at equation number, which one was it? Equation number 11, that is where we had the expression of del mu prime over del of x. So, then from 11, this is what we have, k T del ln C over del of x is equal to minus Z E del psi over del of x. Let this be 14, okay. And if this is 14, then immediately you can realize I can write del psi over del of x is equal to minus k T over Z E del ln C over del of x, right. So, this is the Nernst equation we had derived before, and you derived exactly the same thing again, considering only your flux, which is a combination of both your electrostatic potential and your concentration gradient. I mean the gradient of the electrostatic potential and the concentration. So, this can be integrated further. Respect to x, what I can get is del psi, there is a difference in potentials, which is equal to psi I minus psi O. So, I refers to inside, O refers to outside, okay. I refers to inside, O refers to outside, that means inside the cell, outside the cell, like that, right. So, this would be equal to minus k T over Z E, I can write ln C I over C O, and let this be equation 15. So, exactly the same thing what we had before. So, you see two different approaches, and because you are finally converging at a very fundamental thing, which is the relation between the electrostatic potential or the electrostatic gradient and the concentration gradient, they have to come out to be the same, and that is what you just saw. Okay. Now, where do we go from here? Now, going from here is a little detailed, but I will just quickly show you one thing. Let us consider ion flow through membranes. Okay. Let us consider ion flow through membranes. see both types of ions can flow, positive and negative, right. Both types of ions can flow. Now, if you say that the flux of positive ions is J plus, so this is the flux of positive ions and the flux of negative ions is J minus, then the equation we had before from our previous equation of J C. Can you tell me what the equation number for J C was, which we did in the last class? I will have to figure out again. Not J P, the J C, the next one. Number? Oh, number 10. Okay. No, even number 10. Okay. Well, let's go with that. So, which was well, I'll not put the number out here because I'm just going to write it a little different. So then I can write J plus is equal to minus Z. Now, this is plus because I am talking about the positive flux, E C then U plus, because U plus means I am talking about the mobility of the positively charged ion, right. Then K T over C del C over del of x plus Z plus E del psi over del of x. Okay. So, if this we label as 16, then what will J minus be? J minus will be exactly the same, right, only for J minus, what will happen is Z plus gets replaced by Z minus and U plus gets replaced by U minus. Remember with the corresponding signs though, with the corresponding signs, because Z minus would be having a negative and so on, and then you would get the expression for J plus. Okay. You do these replacements, that is what you would get. Do not make any change in signs, just in place of Z minus put in Z plus, put in Z minus and all those things, right. Now, when you have equilibrium condition, what will happen is, again at equilibrium, anyway it does not have to be in equilibrium, but at equilibrium does not matter whether equilibrium or not, your electroneutrality is always maintained, right. At equilibrium or not, electroneutrality is always maintained, and if that is the case, then from here what I can write is J plus J minus would be equal to 0. J plus J minus would be equal to 0, and then go ahead, which I will not show you, it is just algebra. So, this then we can proceed to finish, but it will give you the final result, and this is what the final result looks like, right. So, again electroneutrality J plus and J minus sum up to 0, and this is what you get. Okay. So, if you go ahead and do the math, this is what you end up with. What you end up with is del psi over del of x is equal to k T over E C del C over del of x. Now, what you have done here is, in this case you have taken Z plus to be 1, Z minus to be minus 1. Okay. You have simplified it, because mostly we are talking about sodium, potassium, chloride and all these things. Now, what is the difference? What is the difference from the Nernst equation you just arrived at, which was equation number, I forgot what it was, 15 or so something. What is the main difference you see out here? The main difference here is it is not only the relation between the potential gradient and the concentration gradient, but this relation is through something else. It is also through the mobilities of the ions, right, because the mobilities of the ions, which is u, they also play a huge role in this ion transport, and it is going to play a huge role because you are talking about diffusion, right. So, if you go and integrate it, that means if you integrate this, if you integrate, okay, this is what you end up with. The del psi is equal to psi of L minus psi of R, where L is the potential on the left side of the membrane and R is the potential on the right side of the membrane. So, this L and R we have discussed before, but that is what it means. Now, this is equal to this equation k T over E u minus u plus. So, this would be this would be u minus plus u plus, same here, this is the mistake, u minus plus u plus natural log C L over C of R. Okay. So, the denominator is an addition and the numerator is a subtraction. Okay. This is your final form of your electrostatic potential difference between the left and the right side of the membranes. Now, how does it help you? So, this is how it helps you. Now, suppose you take a case where positive ions can cross the membrane, but negative ions cannot. Okay, that means the mobility of the negative ion can be taken to be 0, no problem. So, the moment that will happen, that u minus would drop out. If the u minus drops out, you can see what will happen. Let us go back. If the u minus drops out, what will happen? So, u minus are dropping out, you are left with minus k T over E ln C L over C R. What is that? That is your Nernst equation. That is what you just got, is not it? So, the Nernst equation was based on what? The Nernst equation was based on the assumption that it is only permeable to one species of ions. I did not consider all the other ions. I just said either it is sodium, either it is potassium, either it is chloride, does not matter what it is, but this is what it would be. But now I am taking into account, say, another bunch of ions. Okay. So, going forward, if you do a rough calculation, so based on this, that means it is impermeable to negative ions. If C of L is equal to 0.1 molar, C of R is equal to 0.01 molar, then del psi comes to be minus 60 millivolts. So, this is the potential difference you generate because of this concentration difference. Okay. And the second more important thing is electrostatic potential can also depend upon the respective ion mobilities, which is as saw, that is rates of ions, that is what do you mean by that? Means it would depend upon the rates of ion flow across the membrane, and because it would depend upon the rate of ion flow. So, just instead of just calling it as electrostatic potential, why do not we refer to it as a diffusion potential, because finally this form also depends upon the rate of diffusion or the rate of flow, which is the diffusion. So, that is why it is referred to as the diffusion potential. See, the idea behind this is, if you would ever come across these terms, or if you would ever want to think about these resting potentials and all these, which happens all the time in our body. So, this is how you would think about it, and for this you do not have to know any biology. This is you have derived from whatever we have discussed or whatever general thermodynamic principles you know. Okay. Biology is based on this, and that is why you need these equations. I do not know whether you have heard of there is a technique called patch clamp technique. I do not know whether you have heard of it, some of you might have. So, patch clamp technique actually has to do a lot with this electrostatic gradient, with resting potentials and all these things, because you have to find out, for example, what is the potential difference, how we can do that, how they are changing during different signaling events. Okay. So, this is the final equation. Again, we will not derive this, but I just want you to understand the importance of this. See, when we are talking about diffusion, why do we restrict it to only positive ions? We can also have negative ions diffusing. So, think about this, we can be having sodium, we can be having potassium, we can be having chloride. These are the most common ions we encounter anyway. So, that means your membrane would be allowing all these things to pass through, depending upon whatever channels and all these things are opening and closing at any given time. But think about this, your membrane would not be equally permeable to all these ions, right. So, when you are going to try to develop a very robust equation, you have to bring in some sort of accountability in terms of permeability, right. So, that means your equation should be having the permeability of the respective ion through the membrane, as simple as that. That means, you have to make sure to what extent the membrane allows it to go through at any given time. Along with that, the other things fall through. So, this is what the three scientists, Goldman, Hodgkin and Katz, they developed. So, in the presence of several different ions, the equilibrium of the cell depends upon the relative permeability of the ions, and this is what it comes to. So, the V rest, that means the resting potential, that is what V is equal to RT by F natural log. Now, look at the numerator and the denominator. What is P standing for? P standing for permeability. Okay. So, this here, this P stands for permeability. So, Pk would be the permeability of the potassium ion, Pna would be the permeability of the sodium ion, and so on. Okay. Now, concentration of K plus out means outside the membrane. So, look at the numerator again. So, you have K plus out, right. So, this is K plus out, right. Then you have Na plus out, but both of these are going out. So, the negative has to come in, right. That is why you see this Cl minus is coming in, right, because it's referred to as, or it's represented as concentration Cl minus moving inwards. Similarly, in the denominator you see K plus in, Na plus in, and then Cl minus now going out. Okay. So, everything is logical, that is what you have to remember. I do not want you to remember this equation, no, again that is not the idea, but again try to understand where this equation is coming from. You would be having different ions, there is no reason why your cell would be permeable to only one type of ion, and we need the movements of all these ions, right. We need a perfect balance, and hence the one of the most robust equations describing that is this equation. Okay. So, this brings us to the end of diffusion. Hopefully you have not diffused away from the course, right. Now, can you tell me where we started with when we went into diffusion? What did we start with? We started with the protein folding mechanism, isn't it? We started with the protein folding mechanism, then we went to what? We went to the diffusion collision model and we totally diffused out. We came to diffusion, right. Okay. So, maybe it is time to go back, right. So, that is what we will do now, right. So, we will go back and talk about this next major topic, protein folding mechanisms and kinetics. Mechanisms we had started with briefly, thermodynamics we had seen, now kinetics we had not seen at all in that sense, but kinetics is very important and you will soon realize why. So, why in protein folding you need both thermodynamics and both thermodynamics and kinetics going hand in hand. You have to have these to have a very good picture of what is happening. Okay. So, today we will mainly discuss about the nucleation mechanism, but before that I will just do a brief recap. Of mechanisms. So, one of the mechanisms was the framework model. Do you remember the name, the framework model? Good. Now, what happened in framework model was you formed local secondary structural elements, independent of tertiary structure, okay, independent of tertiary structure. So, that means the local elements did not care what your tertiary structure was, it just form those local elements. Remember, these local elements were referred to as those microdomains, right? I understand that it's a long time ago. So, microdomains, if you would remember. Now, what will happen is local elements would diffuse, collide, coalesce or adhere to each other to give you the final native state. Okay. Now, if you look at this brief outline, I have drawn two arrows. One is this and the other one is this. Okay. What does this mean? What it means is this: when you start from the unfolded state, the totally unfolded state, what the unfolded state does is, without even keeping in mind what the tertiary structure might be at the end, it forms local secondary structural elements, right, all these microdomains. So, can you not say then these microdomains are, when they form these microdomains, independent of the secondary structure, and they have not yet diffused or collided to form the final structure? At that point, it is like an intermediate. So, that means you start from the unfolded state, you go to an intermediate, which is essentially microdomains formed independent of the final tertiary structure, and then once you form this intermediate, now this intermediate having these microdomains, they would diffuse and collide, coalesce, because they have to coalesce or adhere in a certain orientation, which is the final native form, to give you the final folded state. That means, this mechanism definitely goes through an intermediate. Okay. So, one of the characteristics of a framework model is the presence of an intermediate. Now, see, it makes logical sense. Why does it make logical sense? Because you are talking about a protein which is it's not one amino acid, right, it is many amino acids, like a polymer of amino acids, and in that case, with all the different conformations available of the side chains and all these things, it is very logical to think about an intermediate being present, an intermediate which would be present on the pathway, and it would help the protein to move along. Okay. That means, you start from a high energy state, which is the the unfolded state, this is the native state, and in between you have an intermediate before this one goes to the transition state. So, the intermediate kind of provides a platform for the other things to happen around it, then you go to the native state. Okay. So, intermediates play a huge role in protein folding. Now, you can ask me what type of intermediates are these. There can be two types of intermediates. One intermediate can be on pathway. What do you mean by on pathway? What I mean by on pathway is, you start from the unfolded state, you are going to the native state, the intermediate you form is a part of that sequence. Okay. That means, you have to form the intermediate to go to the folded state. Then what is an off pathway intermediate? An off pathway intermediate would be, now think about this, off pathway means it has no relation to the pathway, because it is off pathway. So, in that case, if this is your U, right, and this is your N, then your intermediate might be here, because it is not belonging to the pathway anymore. Okay. I will come back to this later, I am just trying to make the reference here. Now, then what the problem is, if you are going to form this intermediate at any given time, and because this intermediate is not on pathway, it will never lead to your final native state, because it is not on pathway, it is off pathway. So, what will this guy have to do? It has to open up, come back to you, and then you has to go to the native state, isn't it, because it is off pathway. Off pathway means, if you are not going along that path, if you are off the track, you are never going to reach your destination. That is exactly what it means by an intermediate being off pathway. So, an off pathway intermediate, if it forms ever, will have to break, come back to the unfolded state, and then the unfolded state again goes to the native state, right. So, you can see it will chew up some time and delay the folding process. Okay. I mean, just keep that in mind. So, essentially what I can say is now that here this would be having an intermediate. And before this nucleation mechanism, almost everyone thought that intermediate is a must for protein folding. Whether we are able to see it or not is a different issue, but intermediates have to be there, okay, because you are talking about such a complicated scenario, right. So, this was about the framework model, and okay, and again to make the point. So, this was the keyword diffusion, where we started our discussion on diffusion, right. And believe me or not, I have not shown you much about diffusion collision model, but if you would be able to if you would try to ever derive an analytical expression for this diffusion collision model, you would start with Fick's second law of diffusion, because it is essentially diffusion of your microdomains. Okay. And then your microdomains can be charged, your microdomains can have distribution of charges, you can put in other electrostatics and all those things, but those are specific ways of looking at it, but this is how people do it and people have done it like this, right. So, that is why we went ahead and discussed diffusion. So, the next one was the nucleation model. The nucleation model. So, here what happened was the protein formed some native secondary structure with the help of neighboring residues, with the help of neighboring residues. Okay. It formed kind of a platform, and immediately what happened was the tertiary structure evolved around it. So, the tertiary structure evolved around it. Okay. So, what we can say is tertiary structure is dependent on the secondary unit. Okay. Tertiary structure is dependent on the secondary unit. Now, what is the difference here between a nucleation model and a framework model? See, how many arrows have I drawn here? I have drawn one arrow, which actually symbolizes that you are not going to have an intermediate. Now, try to realize why. When we talked about the framework model, we found the microdomains were definite type of intermediate, and then it moved on to the final state, which is the native state. Now, look at this. In the nucleation model, you are not saying that you are having full blown microdomains forming. Instead, what you are saying is, if you have this protein, then neighboring residues kind of come together and form a nucleus. That is why it is called nucleation, right. Form the nucleus, and the moment it forms the nucleus, then the tertiary structure evolves around it. That means, the tertiary structure is a result of your secondary structure formation or the secondary structure nucleus formation. Now, all these things are happening at one go, because this nucleation thing is not an intermediate per se. Okay, it does not exist as an intermediate, it is not a well developed intermediate. When it starts to form the protein, what it does is it gets a platform and everything evolves around it in one go. Okay. So, that means what I can say is, then this one is devoid of intermediate, that means no intermediate. But this was a huge issue, right. The issue was, as I just said, that people always thought protein folding to be mediated by intermediates, and if intermediates are not there, which is what the nucleation model proposes, then the nucleation model cannot be there. It cannot be a logical model, as simple as that. But obviously things changed, and that is what we are going to talk about. And the last major one was what? Remember hydrophobic collapse? So, that means protein collapses around a cluster of hydrophobic residues. Okay. So, this is your collapse, there is a collapse form, right. Tertiary structure starts forming, and then what you have is you get the final native state. So, let us think about this briefly. Right now, we are not going to talk about hydrophobic collapse, but how do we envision it? So, for example, you think about a helix, a membrane helix, right, and we have discussed this before. If a helix has to be in the membrane, it better be hydrophobic, right. It has to have a lot of hydrophobic residues, because it has to span across that hydrophobic part of the membrane. So, what will happen is, that means in that part, so if this is your membrane and if you have the helix in this part would be mostly hydrophobic in nature. So, initially what will happen is, the protein will not care. What it saw is that I have a set or a cluster of hydrophobic residues with hydrophobic side chains obviously, and hence it will immediately collapse, right. So, this collapse structure gives you what? The intermediate, because actually it forms a collapse state. Now, once it forms a collapse state, now see what happens, how it is different from the nucleation condensation model. The tertiary structure starts evolving around it, but remember, when we talked about the nucleation model, right, we said that it was already forming a secondary structural nucleus. In the hydrophobic model, we are not saying it is forming a secondary structural nucleus. What we are saying is, it is just forming a hydrophobic cluster, which has no information of the final secondary structure. Once it forms a hydrophobic cluster, the tertiary structure starts forming around it, and after that your secondary structure involves. So, in nucleation model, it was your secondary structure, then your tertiary structure. So, the tertiary structure came only because of the secondary structure. In the hydrophobic model, it is just the opposite. It is the tertiary structure which drives your secondary structure formation. Is it clear? This is what it is, because the moment you remember, the moment you have a collapse is always sudden, and because the collapse is sudden, it is very hard for something to maintain a certain direction to maintain or to form a certain microdomain, because it will always collapse around a certain cluster. See, it is like if I stand, if something happens to me, I collapse, I cannot collapse, and while collapsing I cannot take a certain pose, right. I collapse just like that. It is same for a protein, right. I can, during collapsing, I cannot think that I will fall to my right or fall to my left, I will just collapse. The same thing will happen for a protein, it will just collapse, because it has seen that cluster of aromatic or hydrophobic residues, okay, not aromatic, hydrophobic. So, here obviously then what I can write is, it will go through intermediate, this is important, and the difference is tertiary structure drives secondary structure formation. Okay. Tertiary structure drives secondary structure formation. This is a huge difference from that of the nucleation model. Okay. So, the first and third ones, they go through intermediates, the second one is not going to go through an intermediate, right. Now, groups of well, especially start with one group, group of Alan Forst, he is at Cambridge. What he and his group saw was, they were working on this protein chymotrypsin inhibitor. Okay, chymotrypsin inhibitor, I have written it here. So, this chymotrypsin inhibitor, as you can see, it is an inhibitor, right. So, that means it inhibits chymotrypsin, essentially that is what it means, okay. And it is called chymotrypsin inhibitor 2, that is why it in short form is referred to as CI2. Let us not worry about the function, that is not the idea. You look at the structure. The structure is, well, I will go ahead, this is I have taken this structure from this journal, this is by the group of Forst. You look at this, here this is a disordered loop, right. This is often referred to as the reactive center loop. This is the loop which actually ends up inhibiting the way it goes in, but again you look at this helix. So, it has only one helix and it has how many strands? Beta 4 strands, but look at this again, you go back to what we had discussed long time ago. These two strands, so that means right. So, that means what can I say? Well, actually it says beta 2 and beta 5 too, those are small strands. Anyway, so this strand and this strand are anti-parallel, and what about these two? These are parallel. See, unique, right? This Ci2, the one they worked with is I think a 62 residue protein, or 62 or 64, I forgot what it is. This is a truncated version. The real protein is 83. So, they have taken out just 20 amino acids. Okay. But even in such a small protein, what are the things you have? You have a disordered region. So, this is your disordered region, is not it? This is the disordered region. You have a helical region, you have beta strands forming a beta sheet, but in the beta strands you have both parallel and antiparallel. See, it is unique, right? I mean, so special, just such a small protein, only 62 residues, but it tells it is still having different shades of secondary structure, the types we have discussed, a helix, 2 types of beta sheets, and even the disordered form, it is a well developed disordered form. Okay, just in one protein, one structure, that too a small protein. Okay. So, it says essentially a 4 stranded beta sheet, but they also refer to beta 2 and beta 5 as a small sheet. So, it does not really matter, but what is really important is the mixed parallel antiparallel even in such a small protein. So, anyway, coming back to the main discussion point, what the first group found was that this protein folded through a 2-state kinetics, right. It folded through 2-state kinetics, that means it had no intermediate. If it is 2-state, it cannot be having intermediate, right. And it followed the nucleation model. Now, that is what he proposed. Now, merely proposing would not do, right. You have to do experiments to kind of try to prove this thing. Now, how do you do that? We will try to develop that as we go through the classes, but see, this thing would come to your mind, why was this chymotrypsin inhibitor chosen, right? I mean, there are millions of proteins out there, why did he decide to work on this? One is maybe it was available to him, well that is maybe a small reason, but look at the other reasons why you would need it. One is it lacks disulfide bonds. Okay. Now, think about the simplicity or the complexity of a folding landscape where you would be having disulfide bonds or not have disulfide bonds. If you would be having disulfide bonds, then this mere formation of disulfide bond would chew up some time, because remember when we talked about this Anfinsen thermodynamics, we said that you can just let it form, it will form, or you can allow it to form the disulfide bonds in a wrong way, reduce it and it will come back, rearrange and form the right one. That was the thermodynamic hypothesis, right, from Anfinsen. So, that means the disulfide bonds would be having several different arrangements, you do the permutation, right, and it can form that many different disulfide bonds, but finally it will give you the right structure, thermodynamics will govern, no problem, but anyway it will use up time, right. So, if you do not have disulfide bonds, what are you doing? You are making the path or making the folding pathway simpler, that means you are increasing the kinetics or increasing the rate of folding of the protein. Okay. Now, the second one we have not discussed at all, neither will we, but if you have prolines, you know prolines are very restricted and they have cis and trans isomers. So, generally what happens is in most of the folded forms proline is a trans configuration, but when it starts with a cis configuration. So, that means this cis to trans isomerization would also have to be built in during a folding. That cis to trans isomerization is not fast, it takes time. Okay. So, hence I would rather go for a protein where either I do not have any prolines, but remember I do not have a choice. Why? Because my protein has beta sheet, and for beta sheet you would have to have loops, kinks, and if I have kinks I have to have prolines, right, because that is what. So, I have no other option. The other option for me was to go only for alpha helix, right, if possible, but still I would be having interconnecting regions. Anyway, so prolines would be there, but what they found out was out of the 5, I think 4 of the prolines were already in the trans configuration. So, great, if they are in the trans configuration, then during the folding it will not have to go through this isomerization. So, that means you are making its kinetics or folding pathway simpler, kinetics even or rate even faster. Good, these are 2 major ones. So, this is how you start looking at a certain research problem or trying to define what you are doing based on these things. These are very simple things, but they do affect a lot. Then it has to fold fast. See, if it folds very slow, then you immediately know you would be having an intermediate, because, and I will show you later, because say if it is an on off pathway intermediate, it will go out, it will break, it will come back, and then unity structure would form. If it is an intermediate which is much more stable than your unfolded state, then it would come like this, right. This is your folded state, this is your unfolded state. Your actually energy barrier would increase, and I will show you that, which will end up actually delaying your process. So, it has to be a fast folder. That means you would try to avoid the presence of intermediates essentially. Okay. So, in this case what they found was the half life of the folding was 14 milliseconds. That means you are going from the unfolded to the folded form. Okay. See, 14 milliseconds is fast enough, and go back to the time that Leventhal said it would be if it would go through random search. What was it? Age of the universe, about 27 years or something like that, right. In that respect, 14 millisecond is almost like nothing. Okay. What they found was, I am not going through this, but what they found was, based on different experiments, that structure of the transition state consists of the N terminal part of the alpha helix. You saw the alpha helix, right. It had the N terminal part, this is the transition state, residues 12 to 21, and some extended interactions. Remember, if it is a nucleation model, then I have to have a small secondary structure built around or being stabilized by neighboring interactions, because if I am not forming the full secondary structure, how can it be stabilized by itself, right. It cannot be. If it is a partial secondary structure, it has to be stabilized, that portion has to be stabilized by adjacent interactions, only then can the tertiary structure form around it, right, because if this one is not stable, then the whole thing breaks down like a pack of cards. Okay. So, I have to have kind of a solid platform. And what are these extended interactions? So, the helix is being formed in the transition state. They showed that, I will tell you why how they showed it, and it is stabilized by interactions by these amino acids, which is alanine 16, leucine 49 and isoleucine 57, and these are the side chains. Okay. Now, obviously, the question is, how did they know this, right? I am just telling you facts, I am just telling you experimental facts, but they had to do the experiments to realize that these are the actual interactions involved. Okay. So, then the rest of the protein is weakly structured and is in the process of collapsing around the nucleation site as the ladder forms, and I just told you what the nucleation site is. So, then this is referred to as the nucleation condensation mechanism. So, try to understand the significance of these two words. You are forming a nucleus, a loose nucleus, right, where you do not have fully formed secondary structural elements, and then the whole of the protein is condensing around it, and you put these things together, you have got the nucleation condensation mechanism. So, in that sense, it is a better one than the nucleation mechanism, where we actually do not talk about condensation, but we just imply that this condensation happens. Okay. Now, before going further, I do not think I will be able to come to that in this class, but let us try to think about this. I am talking about a two state folding, right. I am talking about two state folding, in this case for chymotrypsin inhibitor 2. If it is a two state folding, what we know from our detailed discussions before, that it is either can be written as unfolded state in equilibrium with the folded state, or I can write as the denatured state in equilibrium with the native state. See, I am writing it from this side unfolded to folding, because I am looking at the process of folding, right, that is what I am looking at. Now, suppose I ask you that I have to know what the mechanism is. Now, here we are really getting serious. We are actually, whatever we have done before, we are trying to use all these things in visualizing how a protein is folding, and that is what we are doing. If I would try to do that, what I would do is, or if anyone would, see, he would try to characterize both structurally and energetically whatever stuff you have starting from your reactant to your product, right. So, you have to know its energetics, you have to know its structure, you have to know this energetics, you have to know this structure, right, and if there is an intermediate in between, you would have to know that too. But what is the problem with the two state protein? The problem with the two state protein is there is no intermediate, isn't it? So, that means what is the only state I have between my unfolded state and the folded state? What is the only state I have? Transition state, and you know transition state is not easy to deal with, right. So, that means my focus has to be on the structure and energetics of the transition state. TS. Okay. How would you study transition state? I will develop this more on Wednesday, but see, one of the easiest ways of studying transition state is how? By looking at kinetics. Why? What is k equal to? The rate constant equal to from Arrhenius theory? A times exponential e to the minus, or e to the minus, not A times e to the minus E A over R T. What is E A? The activation energy. So, if you would get that activation energy, it is related to the rate. So, it gives you an idea about the difference in energy. Okay. So, that is one immediate way of looking at a transition state. That means you look at the kinetics. That is immediate and one of the easiest ways of looking at, or one of the most direct ways of looking at transition state, because how else would you look at it? Transition state is a transition state. Transition state is very fleetingly populated. Transition state, can you capture a transition state? Probably we cannot. Okay. Now, second, here we are talking about transition states of a protein. Remember, I am not talking about transition state of a simple molecule undergoing a bond breaking. What is the difference? For a protein, and guys really think about this, we have talked about transition state. That means, if you are talking about transition state, we are going to apply transition state theory obviously. This is the chemical kinetics class you looked at in detail, right. But would you really apply transition state to a protein? For example, you take a bond, for example, you take a molecule which is undergoing a transition to go to another molecule, right. So, there few bonds have to be broken for normal molecules, okay, just a molecule, a simple molecule. So, that means in the transition state what is happening is you are actually breaking bonds and also forming bonds. Okay. So, here what you do is you do a quantum mechanical derivation, and if you would remember from your transition state theory, the rate of this breaking or this rate of this transition or the rate constant is dependent upon the vibration of a bond. Okay. You would try to recall that. Okay. But you take this same feature of a transition state and try to talk about a protein undergoing folding from an unfolded state. Are you breaking actually any such chemical bond in a protein? No, right, because see, the peptide bonds are already formed. The only other bonds you have are very weak bonds in the sense you have maybe hydrogen bonds, you maybe have salt bridges, you maybe have these van der Waals forces coming in. In that sense, they are very loose bonds. Okay, so in that sense, you again do not have you do not have a fully developed bond breaking and another bond forming. So, a protein transition state is a lot different than the regular transition state that we all know of. And the last sentence is, when we are talking about a molecule can have its conformations, but the number of conformations would be very limited. But you talk about a protein starts from the unfolded state, it goes to the folded state, it has a huge reduction in entropy, it starts with a zillions of conformations, it ends up in a very few conformations, possibly a one unique native structure. Then what is going to happen to the transition state? The transition state, though it might have many conformations lower than the unfolded state, but still it will be having a distribution of conformations, right. And because it would be having a distribution of conformations, you would think about the top of the transition state, it would not be a sharply peaked one, instead it would be what? A kind of a shallow one, like a shallow transition state with many jiggles. I will show you next class. So, this is why application of transition state to protein folding is an assumption we go ahead and we see that it works very well for us, but in that sense it is not very rigorous based on whatever analysis of transition state or whatever stuff you know about transition state from your chemical kinetics class. Okay. So, it is a good place to stop. On Wednesday we will develop from here, we will look at the transition state equation we use, transition theory, and then see how we can look at the mechanism of folding, because remember, if I do mutations, that mutations might affect your transition state, and if the mutations affect your transition state, your rate will be affected because your activation energy would be affected, and if your activation energy is affected, your rate is affected, then you know to what extent that mutation is involved in the transition state, involved in the folding, and that is how you try to figure out which amino acid is involved. Okay.